Hi everyone. Today we are going to talk about the cell cycle, which is a series of stages that cells go through as they live, grow, and prepare to divide. So the first thing I want you to think about is, why do cells divide? You might want to take a second here, pause the video, and think, what are one, two reasons that we need new cells? Maybe take a second to pause now. Thought about it? Alright, some of the reasons might have come to mind. One reason is simply growth. Many multicelled organisms get bigger over time, and to get that increased size, you need cells to divide to make new cells. Another one, for single-celled organisms, cells divide in order to reproduce. For example, bacteria reproduce in a separate process called binary fission, but they separate one cell into two cells, and now there are two bacteria rather than the one. And one of the more common reasons for division is to repair damaged tissue or to replace cells that have gotten old. Cells do age, and you need to replace them with new cells over time. One thing, before we get too much into it, a very helpful video is the Amoeba Sisters video on this topic. I put a shortened link down there, tinyurl.com/qo mitosis clip. I would recommend checking out that clip, very helpful. They go through the entire process, so if you want to know more, feel free to check out their video. Starting with this diagram here, the cell cycle can be broken down a few different ways. One way is to look at this large blue bar here. Okay? The biggest part of a cell's lifespan is called interphase. This is when the cell is not dividing, when it's doing its job, and then eventually preparing to divide. But cells spend the vast majority of their time in this stage. If a cell is going to divide after interphase, it will enter mitosis, which we are going to focus on today, division of the nucleus, and then last cytokinesis to complete cell division, after which you will have two new cells identical to each other. So here, breaking it down, in interphase, the cell is sort of doing its job, going through the process. It does have to copy its DNA here in order to have two cells. Each one must have the full complement of DNA or chromosomes. Then mitosis, which is division of the nucleus, and last cytokinesis, which separates the entire cell, finishing division. Now, if we just look at interphase is also divided up into stages. Right? The stage that would occur first, because this is a cycle, it's a bit iffy on which stages occur first, but if you're starting with the cell that just divided, the first thing it will go into is G1, or gap 1, and the cell basically grows and does its job. If it's a skin cell, it protects the body. If it's a intestinal cell, it takes in food, does digestion. After G1 is over, now the cell is preparing for division. It goes into S phase. S stands for synthesis. Synthesis means to build. And in this stage, the cell is going to copy its DNA, do DNA replication, which we talked about. After that process is over, DNA has been checked, it's good to go, got an extra copy. The cell will go into the G2 phase, where it will grow further and prepare to divide. It's really committed to division at this point. So what we're going to focus on in this video is basically going through the stages of mitosis and one or two major events in each stage. So once the cell has committed to division, the first thing it needs to start doing is divide up the nucleus. Right? The nucleus, as you know, houses the DNA, and now we have already made a copy of this DNA, but now we must divide it equally and appropriately into the two new cells. Mitosis is divided into 4 stages. Their names are prophase, metaphase, anaphase, and telophase. One way to remember this is the acronym PMAT, and if it helps, you can make it a mnemonic with Pirates May Attack Tonight. But those are the 4 stages: prophase, metaphase, anaphase, telophase. Pro, meta, ana, tela. So we're going to go through those, each one briefly. Starting with prophase here, okay, some of the things that happen in prophase. One, the chromosomes become visible. So back here in picture 1 here, this was an interphase. The DNA is loose, it's coiled up, it's in a form called chromatin. You can't see individual chromosomes. But in prophase, the cell condenses these chromosomes, right, and you begin to see individual chromosomes are apparent. Also, you can see the nuclear envelope breaks down. The nucleus, the surrounding membrane of the nucleus, starts to break down in preparation of splitting the chromosomes. Okay, so here we are in prophase, a thinner nuclear membrane and individual chromosomes, which sort of look like thick X's. We'll talk some later about chromosome number and organization. So after prophase, we're going to move into metaphase. Okay? One way to remember metaphase is M for metaphase, M for middle. The chromosomes line up in the middle of the cell. The spindle fibers, which are these things here, they're attached to the chromosomes and they move them into the middle of the cell in preparation for splitting. So here we are in metaphase, everything's lined up nicely in the middle there. Okay. Moving to anaphase, the 3rd stage of mitosis. Okay. Another way to remember anaphase, A anaphase, A away. The chromosomes split apart, so we say the sister chromatids move away from each other. So before they looked like an X, now it looks like half an X. So basically they split it apart, split it in 2. Okay, 2 identical copies that were attached now are pulled apart. Okay, so here we are in anaphase. Now, a note about chromosomes and chromosome organization and naming. A chromosome, before it's copied, is basically just a strand of condensed DNA. After it's copied, that's where you sort of get this X shape. It's 2 single-stranded chromosomes that are now attached, so it's a double-stranded chromosome. There's a lot of trickiness understanding the naming and numbering of these chromosomes, because this down here is a chromosome and this up here is a chromosome. This is a single-stranded chromosome, this is a double-stranded chromosome, but they're still just called chromosomes. But now 1/2 of this double-stranded chromosome is called a sister chromatid. So these are identical copies. So this is identical to this, barring any sort of mutation during replication, but generally identical copies. Okay, and these are created during replication. So if you remember, that occurs during the S phase of interphase. So before mitosis has started, these have all been copied. All right, on to the 4th and final stage of mitosis, telophase. Okay. Again, a way to remember, telophase, T, 2 nuclei are visible. So now we're starting to get where it looks like 2 cells instead of 1. New nuclei start to reform around the chromosomes. Okay. The cell membrane will pinch in an animal cell, okay, whereas in a plant cell, they start forming a structure called the cell plate. Because plant cells are surrounded by a rigid cell wall that won't pinch in, they build a cell plate going down the middle in order to separate their cells. Now what really matters here is, what do you get out of mitosis and why do we need to do it? So we talked about why mitosis happens: growth, repair, and reproduction. But what do you get out of it? Okay, so 2 cells come out, whereas 1 went in, and they are genetically identical to each other. Okay, again, barring mutation errors, but genetically identical. They're not significantly different. Generally, their DNA in one cell, the DNA in one cell is identical to the one in the other cell. Okay, the daughter cells have the same number of chromosomes when compared to the parent. So for example, if the parent cell had 40 chromosomes, each of the daughter cells have 40 chromosomes, and they are the same 40 chromosomes. They are identical to each other and the parent cell. And the daughter cells are diploid. The prefix di means 2. So what this means is the cells that come out have 2 sets of each chromosome. So for example, chromosomes are generally just named by a number. So for example, chromosome 1, the cell will have 2 copies of chromosome 1. It'll have 2 copies of chromosome 2, 2 copies of chromosome 3, 2 copies of chromosome 4, and so on. Right? So that's what diploid means, as opposed to haploid, where a cell only has 1 copy of a chromosome. Um, Number of chromosomes varies dramatically in different species. You should know that humans have 46 chromosomes, or 23 pairs, but different species have different numbers. For example, fruit flies have 8 chromosomes. This is one of the reasons they are widely studied in genetics, because they have a smaller genetic code and it's easier to track changes in them. Kangaroos have 16 chromosomes. A pineapple has 50 chromosomes, so a little bit more than we do. The New Caledonian crow has 80 chromosomes. A carp has 104 chromosomes. And a hermit crab is one of the animals with the highest chromosome number. It actually has 254 chromosomes, or 127 pairs. Chromosome number doesn't appear to be correlated to any really specific trait. It's not good to have more or less, but it is interesting how species got such widely different numbers of chromosomes. One thing here, after mitosis, division is not done. We've divided up the nucleus, okay? We have partially pinched in the membrane, but we haven't finished division. So we need one more stage to happen, and this is called cytokinesis. Cyto means cell, and kinesis means to move. Okay, basically we're going to finish division here and create 2 separate cells. Cytokinesis, the cytoplasm divides, meaning everything besides the nucleus. All those organelles, you need the mitochondria, Golgi, etc., lysosomes, they need to go into each of these new cells. 2 daughter cells will form. At the end, you've got 2 daughter cells. These structures here, I meant to mention, are called centrioles, and they were where the spindle fibers connected that pulled apart the chromosomes. And then after we mentioned this, after division, these cells are genetically identical to each other and to the parent. Now we have gone from 1 cell to 2. One note here about cells and cell growth is a famous cell line is called the HeLa cells, and they're an immortal cell line used in research. When we say immortal, what that means is most cells, if you try to culture them in a lab, like grow them, they will only divide so many times before they are no longer viable, or they will die, or they won't function properly. But we are able to turn these HeLa cells into an immortal cell line where even after many, many divisions, they would keep dividing and keep living and just continue to divide. These cells are called HeLa cells because they were taken from a patient named Henrietta Lacks. So they took the first 2 letters of first and last name from a tumor from her. And actually, it's a very interesting story about they took these cells without her permission and were using them for decades before her family even knew. So if you're interested in that, I would suggest looking at this book, The Immortal Life of Henrietta Lacks, a really interesting story about these kind of cells. That basically does it for our explanation of cell cycle today. We wanted to hit the basics of the different stages and the stages of mitosis and why this process matters. This is how you have gone from the 1 cell you started out as to the about 30 trillion cells you are now, doing this process over and over and over. Again, that video I mentioned at the beginning is very helpful, so if you would like more information, I would suggest checking it out. The Amoeba Sisters video is at tinyurl.com/qomitosis clip. That's all I have for today. I hope you found it useful.